The next speaker is Professor Fred Chow from the Australian National University. Professor Chow is an emeritus professor at the ANU, and he started his career as a postdoc at Prof Jim Barber's lab. So without further ado, let us welcome Professor Chow.
Thank you. When Berto came to the ANU earlier in the year, he said Jim will be getting an honorary doctorate and said that it would be nice if I could come and join in the celebration. So here I am, and thank you to the organizers for inviting me to speak. What I would like to talk about today is actually all work that I took part in when I was in Jim's lab on one aspect of natural photosynthesis. It's to do with the electrostatic repulsion that determines or helps determine the ultrastructure of the chloroplast. So my journey to Jim's lab started back in the late 70s. I started my PhD, with Alex Hope, who knew Jim from way before that. And when I was writing out my thesis, Jim offered me the postdoc, but I couldn't join his lab because I was stateless at the time. I needed to stay in Australia in Boardman's lab to get a passport before I could travel. So I joined Jim's lab a bit later in '79. And then I eventually ended up back in Australia via Littlehampton, the Glasshouse Crops Research Institute, joining Jan Anderson's lab, where Berto had spent some years before I joined the lab. So I want to talk about grana, the structures that are formed in the chloroplast, consisting of stacks of thylakoid membranes. Each thylakoid is like a hot water bottle with many necks interconnected to other thylakoids, and they form a stack or granule stack. And in some plants, they can be quite big, and this particular one from Alocasia grown in low light has over 160 thylakoids in one granule. And it's the electrostatic repulsion that can influence the size of these grana. So because of the formation of the granule, the distribution of the membrane protein complexes can be quite heterogeneous, something that Berto and Jan Anderson famously worked on in the early 80s onwards. So for example, photosystem 1 with its protrusion and the ATP synthase, they cannot be accommodated within the so-called oppressed membranes because this partition gap cannot accommodate the protrusions. So these complexes are located in the non-stacked regions of the system. On the other hand, photosystem 2 doesn't have much in the way of protrusion, and they tend to be concentrated in the oppressed membranes. The Sargum BF complex has only a small protrusion, and so it can be pretty well located in stacked and non-stacked membranes. So, well, grana are not really essential for photosynthesis, but they do help to fine-tune the process. So they, for example, they allowed the system to optimize the packing of the pigments, avoid too much spillover of energy from one photosystem to the other, and thereby limiting the waste. It can regulate safe dissipation of too much light energy. And it helps to protect non-active PS2s from premature degradation of one of the components. And something that I discovered is that under identical conditions, stacked membranes can perform non-cyclic electron transport-driven ATP synthesis at 2 or 3 times the rate compared with unstacked membranes when assayed in the same medium. So there are various forces that together determine how much stacking takes place. There would be the repulsive forces. The obvious one is the electrostatic repulsion because the membranes carry net negative charge on the surface. And there's hydration repulsion associated with the structured water and near the, at the membrane surface. And that prevents too close a contact between adjacent membranes. And there's, of course, I've touched on the steric hindrance due to protrusions of these membrane complexes. So these are all repulsive. And then there are attractive forces, the van der Waals force of attraction, and there are also entropy-driven attractive forces. But mainly today I will talk about the electrostatic repulsion, and that's what attracted me to Jim's lab to work. And calculation of the electrostatic repulsive force requires a knowledge of the surface charge density, the net surface charge density, coulombs per square meter. This year, a group headed by Helmut Kirchhoff at Pullman published a paper in Nature Plants where they summed the exposed charges, negative and positive, on proteins and lipids, and arrived at a figure of a negative 39 millicoulombs per square meter. And we measured back in the 1990s a value which was about slightly less negative than that. So, and we used a divalent cation, methyl vialogen, and measured the amount of it sequestered in the diffuse layer and arrived at a figure like this for spinach thylakoids. For barley, we used another related compound, diquat, labeled C14 labeled diquat, and obtained slightly lower negative charge densities on barley thylakoids. And the group in Pullman recalculated the van der Waals force of attraction as well. And they found, encouragingly, that the recalculated attraction is now 4-fold larger than was previously reported by Scully and others from Canberra. And they calculated the electrostatic repulsion based on this calculated surface charge density and concluded that the van der Waals force of attraction was sufficient to overcome the electrostatic repulsion without having to invoke any entropy-driven attraction. So just the van der Waals force was sufficient to overcome the repulsion. And this is what they found. I picked 2 panels from the figure. This is just looking at the magnitude of the forces. First of all, the electrostatic the hydration repulsion declining with distance from the membrane surface very rapidly. Then the van der Waals attraction, the magnitude of it, declining in this fashion. And these 2 curves are reproduced in the lower panel. The electrostatic repulsion based on this calculated surface charge density without any phosphorylation of the proteins varies like this for this ionic composition. Of course, it does depend on the ionic composition of the medium. At 5 millimolar magnesium and 20 millimolar potassium, for example, the repulsion is still greater than the attraction, so this not expected to be too much stacking of the membranes. But when you increase the monovalent cation concentration to 100 millimolar on top of the 5 millimolar magnesium, now the two are pretty similar according to their calculations. And if the monovalent cation is increased further to 200, then the red curve will be well below the van der Waals attraction curve. So, in other words, increasing the monovalent cation concentration decreases the electrostatic repulsion and should promote stacking. But actually, that's contrary to what we observed experimentally. There's a discrepancy then between their calculations and our experimental observations, which I will come to. We observed an antagonistic effect of divalent and monovalent cations on the extent of thylakoid stacking, but that was not predicted by them. So first of all, we had to have a way of quantifying the extent of stacking using a method that can be applied to a large population of thylakoids and in a convenient fashion. So what we did was to break up the stacks from the connecting non-stack membranes by either detergent treatment, or we can also use sonication. After the treatment, we have the chunks of the granule stacks, which we can centrifuge and collect in the pellet. So then it's a simple matter to collect to measure the proportion of the chlorophyll that comes down in the pellet after centrifugation for at 10,000 G. And so this is what we see as we increase the magnesium concentration for any given background of monovalent cation concentration. There's increase in the extent of stacking. This is the observation. And there's this antagonistic effect that I mentioned before. At 5 millimolar magnesium, actually increasing the potassium chloride decreases the stacking, which is not predicted by the work from Pullman. And this is confirmed by measurement of chlorophyll fluorescence, which was done in the presence of DCMU, basically monitoring the extent of spillover of excitation energy from photosystem 2 to photosystem 1. So it's a real effect that we observe crossing over of the two curves. And to predict this antagonistic effect of divalent and monovalent cations at the known thickness of this gap between two opposite membranes of about 3.5 nanometers. Barry Rubin, myself, and Jim considered the outer Helmholtz plane back in 1981. And this is the outer Helmholtz plane. That's the membrane surface with the net negative charges on the surface. And there would be a layer of solvent molecules adhering to the surface, and occasionally there would be some cations that are partially solvated and are adhering to this membrane surface. And the diffuse layer is out here, where the monovalent cations can diffuse freely. And anyway, when we use a monitoring cation like methylvalogen with two positive charges, the cation is monitoring the diffuse layer and sees a certain surface charge density that is behind this outer Helmholtz plane. So any binding of specific binding of cations would then cancel out the charge as seen by the monitoring cation. So since sigma is measured by the sequestration of a screening cation such as MV2+ in the diffuse layer, the value that we obtain refers to the net surface charge density at the outer Helmholtz plane, beyond which ions can diffuse randomly. That is, any specific binding of the partially dehydrated cations, such as, say, sodium to the negative membrane behind this outer Helmholtz plane, will give a lower magnitude of the charge density as seen by MV2+. And this may explain the larger calculated magnitude of the surface charge density than measured experimentally. So perhaps I come back. So the as far as the methyl divalent cation is concerned, the surface charge density actually starts somewhere here. In other words, the surface potential starts here, whereas the curves for the hydration repulsion and the van der Waals attraction, they start from the membrane surface. Because of this, in the 1981 paper, we actually transposed the electrostatic repulsive forces. The curves are transposed so that the surface potential that was measured starts at this separation between the two opposite membranes. At this separation is 2 times the distance from the surface to the outer Helmholtz plane. So that is 2.5 nanometers or so. That's where our surface potential starts. So because by transposing these curves across, the magnitude of the repulsion is now much greater than the attractive forces, the attractive force. And using that idea, we could then predict the repulsion between two membranes, the magnitude of the repulsion, force per area, and we could predict this crossover effect, the antagonistic effect between monovalent cations and divalent cations that I talked about. So we transpose the repulsive force so that the surface potential corresponds to the charged surface, the outer Helmholtz plane, with the charge density sigma as seen by this divalent cation. By contrast, the curves of attraction and the hydration repulsion begin at the actual membrane surface. Because of the transposition, the electrostatic repulsion greatly exceeds the attractive force. The conclusion would be that we still do need the entropy-driven attractive force, which I'm not discussing in this talk. It's this entropy-driven attraction which, together with the van der Waals attraction, that can overcome the electrostatic repulsion and bring the membranes to the equilibrium distance of separation of about 3.5 nanometers. And thank you for your attention. And just to this is a picture taken back in 1994 when Jim came to Canberra for a conference. And that's another conference in 1998 when we celebrated Jan Anderson's official retirement at the conference in Arnsberg, organized by Barry and Cornelia Osman. Thank you.